Welcome back, everyone. We will now begin session one on diversity conservation. And the first speaker of the session is Sanya Gupta, who will give a talk on in vitro propagation and conservation of Atrocarpus lacucha, an underutilized fruit of India.
Good morning, everyone. I'm Sandhya Gupta from ICAR, National Bureau of Plant Genetic Resources. I'm going to present my work on in vitro propagation and conservation of Artocarpus lacucha. This work was done by Vivek, a graduate student. At the outset, I also want to thank Professor Kanchit Thamasiri, who has given me opportunity to present this work. Artocarpus lacucha is very important species native to India. It is popularly known as monkey jack, lakuchi, barhar, lakucha in India, padahar in Nepal, mahat in Thailand. It is distributed throughout the Indian subcontinent and Southeast Asia. On the left side of the map, you can see different regional stations which are working under ICAR, NBPGR, which is my institute, and the genetic diversity of Artocarpus is conserved at the regional station Ranchi, which falls at the eastern part of the country. Artocarpus lacucha is also very known for medicinal purposes in Thailand. The left side below, the distribution map of lacucha is shown in Thailand, where the yellow dots are showing the distribution of lacucha and the pink stars are showing another species distribution across the country. Artocarpus lacucha belongs to family Moraceae, a family which has jackfruit in it. It is a multi-purpose tree. The tree is valued for furniture, timber, or animal feed, apart from the edible fruits. The timber of lacucha is durable and can be compared to teak wood. That is why it is preferred for making furniture and also used for construction work. It is a monoecious species where the male and female flowers are borne separately on the same tree. The fruits are sweet and sour in taste. Ripe fruits are eaten fresh. Fruit pulp is believed to be a tonic for liver. Raw fruit and male flowers, they are used for chutney and pickles, and it is a seed propagated crop. Population of lacucha is generally decreasing because the trees are being used for construction, wood, fodder, and timber. So they are being cut. Seeds are very poor in viability, so regeneration is very low from seed. The seed viability of lacucha is very low, as I said. That is why propagation affects. And vegetative propagation methods of lacucha has not been successful yet. That is why in vitro micropropagation protocol for rapid multiplication of lacucha is required. To maintain adequate supply of plant to establish new plantation and to conserve the desirable genotypes. Keeping in mind, this study was taken up to study the effect of different hormones on in vitro shoot parameters and regeneration of Artocarpus lacucha, and to study various methods under the in vitro conservation, such as type of enclosure, storage temperature, and inclusion of mannitol in medium. Micropropagation is very important and it is required for mass multiplication. And for in vitro conservation, there are several methods that we have used: normal growth and the slow growth strategies. The in vitro cultures for Artocarpus lacucha, which were maintained in the culture in the gene bank, they were used for further studies, but the initial plants which were used for raising the cultures were from the tree grown in the campus. Sterilization was done as per standard procedures. This slide shows different concentrations of media. Basically, two cytokinins were used, metatopolin and BAP, in different concentrations to see their effect on growth parameter. Mannitol in different concentrations was used for in vitro conservation. Cultures were kept at culture room, standard culture room conditions, except those which were for to see different temperature effect, they were kept at different temperatures in the BODs. Statistical analysis was done as per standard procedure from the computer-based software. So we have seen the BAP and metatopolin, they were very effective for shoot multiplication, shoot growth, shoot length, and number of node. These are the cultures which were on BAP, and these are the cultures which were on metatopolin. The number of shoot varied from 3 to 6 in BAP and 3 to 5 in metatopolin. Higher the concentration of any cytokinin, whether it is BAP or metatopolin, it showed a lot of callus. And increasing the concentration is also showing the reduction in shoot length and the shoot number. BAP at 5 degrees, 0.5 mg per liter was optimized, where we could get 5.42 centimeter of shoot, 5.42 centimeter of number of shoots were 5.42 and the shoot length were 2.25 and the number of nodes were 2.48. The plus point was here that there was no callus. Here you can see in metatopolin in higher concentration there was callus, and here again BAP at very high concentration there was high amount of callus. We do not prefer callus at any stage of micropropagation or in vitro conservation. You see the effect of different temperature on the cultures of Artocarpus lacucha. We have used 5 degree, 10 degree, and 25 and 5 alternate temperature, and this is 25 plus minus 2 degrees centigrade. At 5 degrees centigrade, in 6 months, the cultures were etiolated and they did not germinate. While transferring on new fresh media, at 10 degrees centigrade, their cultures did not show any growth at all. But at alternate temperature, shoot multiplied, they grew, but the leaves were retarded. They did not, there was no leaf growth. In culture room temperature like 25%, that is a normal growth temperature. The cultures were growing. So we can say these 25 degrees centigrade is the temperature where the cultures could be kept for conservation. The advantage of normal growth temperature is that we do not need any BODs or low temperature incubation equipment. You see, effect of culture tube enclosures. We can see when we are using these cotton plugs, then the media got depleted. The cultures were also showing less growth. There was no leaf. So the difference was remarkable. Here the media was intact, and I'm sure these cultures will continue in the same stage for next 6 months or so without subculturing. Then we have tried to see the effect of mannitol in different concentration. And for 6 months, the growth was fine. There was no callus and there was no morphologically abnormality would be recorded. But again, we have to see in long run whether these cultures will be in the same good health or the health, or they show any abnormality. So to summarize, we can say the metatopolin, out of metatopolin and BAP. BAP was better for shoot parameters. For in vitro conservation, the cotton plugs were not good, but the cultures capped under the screw cap, they were good up to 6 months. And the temperature, culture room temperature, the 5 and 10 degrees are not supporting this plant growth, but the 25 degrees centigrade was okay. But if we have the incubation, low temperature incubation, then 25 and 5 degree alternate temperature can be seen for conservation. Otherwise, 25 plus minus 2 degree centigrade looks good. And for mannitol, again, we have to, the experiments are still continuing. And these shoots were also grown for rooting, to see the rooting in there, and then these will be transplanted into the pots for hardening, and we'll see that how many cultures survive. I again want to thank Professor Kanchita Masiri, convener of this conference, and the organizers to giving me opportunity to present my work, Director ICAR and BPGR Corolai, for permission, and the staff of TCCU to facilitating this work.
Thank you. The next talk will be by Vichai Buribanya Wanit on native Thai water lily survey as an evidence to support Jongkoni water lily existing in a new subgenus.
Good morning, every participant. My name is Vichai Puri Panjawanit. Today, I am pleasure to present the topic of Thai native water lily survey as an evidence to support Jokoni water lily or Nymphaea siamensis exists in new subgenus. In Thailand, there are 4 native and 2 imported species of nymphaea have been recorded. The native are 1. Nymphaea cyanea, 2. Nymphaea nuchari, 3. Nymphaea pubescens, and 4. Nymphaea rubra. And the imports are Nymphaea capensis and Nymphaea colorata. Before Nymphaea siamensis was given a scientific name, explorations were taken around Thailand to investigate native water lily which might be related to Nymphaea siamensis. Some of them were used as evidence for scientific nomenclature of Nymphaea siamensis, but some data were not previously published. A lot of variety details were interesting in many aspects. Water lily samples were collected from all around region of Thailand, from the north to the south and from the west to the east. Pusa Dok Baw National Park, Nong Han Lake and wetland area in northeastern, Sarapi district in Chiang Mai in the north, Thale Ban National Park and Thale Noi in the south, and also Pangabon Water Lily Garden and wetland area in the central plain of Thailand. Morphology and taxonomy studies were done at Green Slipit Botanical Garden, Chiang Mai, Thailand. Let's start to consider Thai native water lily. บัวขาบ or บัวขาบภาคกลาง or Nymphaea cyanea. The name บัวขาบ might refer to many types of water lily which are in the shade of light blue or purple blue. The Thai native people in central region call บัวขาบ or ขาบ water lily. บัวขาบ or บัวขาบภาคกลาง flower in this picture. นิรุบล or บัวแบม่วง It looks like บัวขาบ or ขาบ water lily with light blue or blue purple petal shade. But after RAPD DNA fingerprint was performed, it was determined that บัวแบม่วง นิรุบล was far different from บัวขาบ. บัวแบม่วง นิรุบล at 101 in the northeastern part of Thailand. This is me in this picture, you see. Nilobon or บัวแบม่วง has morphology similar to บัวขาว but DNA fingerprints was far different from บัวขาว บัวแบกขาว บัวแบกขาว water lily has morphology very similar to บัวแบม่วง. It's found in Mukdahan in northeastern part of Thailand, and RAPD fingerprints show that บัวแบกขาว was close to บัวแบม่วง and close relation to Nymphaea siamensis too. บัวแบกขาว water lily flower. บัวผัน and บัวเผื่อน or Nymphaea nuchari are also well known in Thailand. The petal is pale blue purple with blue white sepals. The petal change its color to pink after the second day of blooming. บัวผัน flower has stellate shape with the size of 5 to 13 cm, while บัวเผื่อน has also stellate shape with the size less than 5 cm. บัวผัน and บัวเผื่อน are similar. Only one thing different. บัวเผื่อน has flower size smaller than บัวผัน. บัวสาย in Sarapi district, Chiang Mai in the north of Thailand. We found that บัวสาย in Sarapi district show distinctive characteristics. Its petals were not totally white, pink, or red like other บัวสาย. The flower exhibits small white petals with a pale pink lip. In addition to Sarapi district, Chiang Mai, it was also found in some other areas of Chiang Mai and Lamphun province nearby. บัวสาย in Sarapi district, Chiang Mai flower. This me in this picture also. Bosa in Salapi district. I think it's very beautiful. Bosa in Pusa Dok Baw National Park, Yasothon, in the northeastern part of Thailand. The flower of Bosa found in Pusa Dok Baw National Park show very pink shade on the outer side of that petal, but the inner side was white. It had rarely been found anywhere else and now is facing extinction. Bosai in Pusa Dok Baw National Park, Yasothon, northeastern part of Thailand and its habitat on the top of the mountain. This picture shows Pusa Dok Baw National Park authority who came with our research group. Bonsai in Pusa Dok Baw National Park were collected to plant in the pot plant. Bonsai in Thalenoi Station, Pathalung Province, in the southern part of Thailand. It looks similar to bonsai from Pusa Dok Baw National Park and bonsai from Sarapi District, Chiang Mai. The pink-white bonsai water lily in Thalenoi was very highly specific in its habitat. It was found only in a small location, which was called Got Nang Yuan and nearby area. Bonsai water lily exploration in Thalenoi Station, Pathalung, in the southern part of Thailand. The pink-white bonsai in Thalenoi Station was very beautiful in my opinion. Bonsai water lily in Thaleban National Park stood at the most tip of the southern part of Thailand. Bonsai water lily from Thaleban National Park had a long pink stripe running from the top to the bottom of their petals. This national park close to the border of Thailand and Malaysia, so this variety may be found in Malaysia too. Bonsai water lily survey in Thaleban National Park stood at the most tip of the southern part of Thailand. Also bonsai water lily in Thaleban National Park stood. This is the picture of native pink night blooming water lily in all over Thailand from our survey. This is a native white night blooming water lily in all over Thailand from our survey too. One type of bicolor mutation. This is a pink white bicolor in native night blooming water lily in Thailand. So beautiful. Let's go to our main story about the Jongkoni water lily. The scientific nomenclature of Jongkoni has recently been accessed from the publication of Nymphia siamensis, the new species of water lily in Thailand, in Acta Horticulture Journal number 1035 in May 2014. Nymphia siamensis or Jongkoni is the historic water lily of Thailand. We can say that it's unique of Thailand. Dr. Sarla Vasuwat, a Thai water lily guru, described Nymphia siamensis or Jongkoni water lily characteristics and history in his two books. One, Encyclopedia of Ornamental Plants of Thailand, and two, Culture of Ornamental Water Lily before its nomenclature. Let's see this chart. Genus Nymphia has 6 subgenus in 2 groups, Apocapriae group and Syncapriae group. In Apocapriae group there are subgenus Anephia, Bacillus, Confluentes. And in Syncapriae group there are 3 subgenus too, Hydrocalyx, Lotus, and Nymphia. And now Nymphia siamensis or Jongkoni now is in subgenus Bacillus in Apocapriae group. Let's notice one thing must recognize. We notice that Nymphia siamensis has no sexual reproductive organ. Propagation is only by bowlet. And what are bowlet? Nymphia siamensis lack both stamen and pistil. Its reproduction is only via bundles which are attached to the vertical rhizome. Usually, this bundle will give rise to leaves. However, when separate from the main rhizome, they will give rise to the new plant. Okay, you can see this is bundles. The unique characteristic of Nymphia siamensis. In addition to Nymphia siamensis, from our previous research, we discover only two water lily varieties have bundles as Nymphia siamensis. The first one, Nymphia nilobon variety and Nymphia colorata. This is a new data in water lily world. From every water lily sample we collect around Thailand, north to south and east to west, from cluster analysis DNA data, we found that DNA fingerprints of Nymphia siamensis is grouped together with Bobam muang nilubon and far from Nymphia colorata. So, with the reason of one, Nymphia siamensis jongoni has no lockroot. Nymphia siamensis jongoni propagate via only bundles and no sexual reproductive organ. 3. Nymphia siamensis DNA fingerprint data group is separate from the other known species in Thailand but very near with native Bobam muang variety. Maybe Bobam muang is a new species nomenclature request in the future. Therefore, from the data, Nymphia siamensis was established as a new species and given the name Nymphia siamensis in 2014. Nymphia siamensis was established as a new species in 2014. Here, this is Nymphia siamensis report. Official accept as Nymphia siamensis. Official scientific name of Nymphia siamensis, V. Puri Panyawanit. V. Puri Panyawanit is my name. Let's back to Nymphia, genus Nymphia chart again. Let's notice that Nymphia in genus Nymphia has 6 subgenus in 2 groups, Apocarpiae and Syncarpiae group. Apocarpiae and Syncarpiae group was distinguished by Syncarpus carpel or Apocarpus carpel in their locule. Please observe that Nymphia siamensis has no locule. So, how to contain Nymphia siamensis in Apocarpiae group or Syncarpiae group? It should be contained in non-Carpiae group in the new subgenus, isn't it? Let's back to see the rule to categorize the taxonomy of genus Nymphia. Genus Nymphia is categorized into groups with a total of 6 subgenera, where the 2 groups are distinguished by local characteristics. Apocarpiae group, Apocarpus ovary consisting in separate carpel characteristics. There are 3 subgenera in this group: Annephila, Brachycellus, and Confluentres. Syncarpiae group. Syncarpiae group Syncarpus ovary consisting of united carpel characteristics. There are also 3 subgenera in this group: Hydrocalyx, Lotos, and Nymphia. Nymphia siamensis has previously been placed under Apocarpiae group in subgenus Brachycellus. It is correct. Nymphia siamensis had no ovary, no locule, no carpel, no pistil, and no stamen. In fact, Nymphia siamensis should be introduced to a new subgenus in non-KPA group. This is my conclusion. And acknowledgement. The first one, our consultant. We thank you to our consultant, Dr. Servla Vasuwat, Thai pioneer, water lily specialist. And the second is the National Research Council of Thailand, who grants fund to our research group. Thank you for your attention.
The next speaker is Reshma Ranjanan, who will give a presentation on agromorphological and genetic diversity of Indian large cardamom cultivars, Cardamomum subulatum, in the sub-Himalayan tropics.
Good morning all. I am Reshma Ranjanan from Indian Cardamom Research Institute, Kerala. Our present study is on agromorphological and genetic diversity of Indian large cardamom cultivars in the sub-Himalayan tropics. Amomum subulatum is commonly called as large cardamom or black cardamom, which is a highly valued spice. The dried ripe fruit is the spice of commerce and having similar properties of those of Elateria cardamom. Elateria and Amomum are the two genus, comes under the same family, Zingiberaceae, and commonly called as cardamom. Elateria has more intense flavor, while Amomum has a camphoraceous flavor and more pungent in nature. And large cardamom is cultivated mainly in Nepal, India and Bhutan. Nepal is the largest producer of large cardamom and India is the second largest producer. During the last year, India produced 8,600 tons from 43,600 hectares and export value yielded 6,759 lakhs from 1,100 tons. The primary chemical constituent of cardamom oil are alpha-1,8-cineol, alpha-pinene, beta-pinene, etc. The morphological and molecular marker studies would be a step forward in defining descriptors. Descriptors are not yet released for this crop. The molecular markers are the solution to evaluate genetic diversity at the DNA level, and the outcome can be effectively applied for breeding and integration of new alleles from unutilized germplasm accession. Habit and habitat of Amomum. Amomum is indigenous to eastern Himalaya and is a monocotyledonous perennial herb with the subterranean rhizome. The aerial leafy shoot ranges from 50 to 140 number and the shoot height ranges from 1.5 to 2.6 meter. And each tiller having 9 to 13 leaves. The leaves are glabrous with a prominent midrib. Inflorescence is spike. In each spike has 10 to 15 fruit. Fruit is a capsule and trilocular with the many seeds. The optimal climatic conditions are altitude ranges from 800 to 2000 meter above mean sea level and the precipitation ranges from 3000 to 3500 mm and temperature ranges from 6 to 30 degrees Celsius. Then objectives. First objective is to assess the morphological and genetic diversity among the natural cultivars and the released varieties of Amomum subulatum, and to study the genetic variability and relationship between the genotypes and to identify the core genotypes. Coming to methodology, the primary step is plant sampling and morphological characterization and molecular characterization. For morphological characterization, Amomum subulatum characteristics shortlisted and modified as per small cardamom descriptor and dust guidelines. In molecular characterization, 20 ISSR and 10 SSR primers used, and the DNA is isolated from the selected plant samples and its quantity and quality analyzed using nanodrop spectrophotometry feature and agarostil electrophoresis, and it is followed by scoring and data analysis. For data analysis, NDS-5 software used. Total 10 genotypes are selected for this study. Two high-yielding released varieties. First one is ICRI Sikkim 1 and the second one is ICRI Sikkim 2, and the 6 natural cultivars Savni, Vorlanki, Holse, Saramna, Ramsi and Ramla, and the 2 outgroups. First one is wild Amomum species and Elateria cardamom. For plant sampling, first morphologically distinct cultivars are shortlisted from the sub-Himalayan tropics, and all the plant samples were collected from the different locations of Sikkim. This geographical map showing the sampling points of Amomum sublatum. One outgroup varieties collected from ICRI Mayiladupara, Kerala. Coming to the result, morphological characterization. The significant differences was observed among the varieties with respect to the number of capsule and spike. Palangi had the highest number of capsules per spike, followed by Ramla and Savni. The varieties also showed a significant variation in weight of the capsule. This obvious that fresh and dry weight of the capsules varied significantly. The cultivar Savni shows higher number of productive spikes while comparing to the other cultivars. Varlangi had attained maximum plant height compared to other cultivars. In all other varieties, the plant height was less than 2 meters. Then this is the classification of morphological characters modified as per small cardamom descriptive and dust guidelines. Total 23 characters shortlisted, which includes morphological, reproductive as well as yield characters. This picture represents the morphological diversity in large cardamom cultivars, and it is highly diverse. And this is the chiller color. They are wearing light brown, light green, and brown red. And the shape of the leaf also shows variation in different cultivars. First one is lanceolate or brown lanceolate, elliptic, and ovate. The primary growth habits also showing 3 different patterns. First one is erect and semi-erect and drooping. And morphology of the capsule shape also very conical. They are conical, ellipsoid, ovoid, cylindrical, and globose. Color of the mature fruit and the spike color is also varying. Flower characteristics also vary with the different cultivar. This is the flower characteristics and diversity of different cultivars. Varlangi, Ramsi, Saramna, Sane, Golse, and Ramna. And this is the spike diversity observed in different cultivars. Spike length, color, and breadth also vary. Then correlation of morphological characters. The highest correlation was observed in between number of capsules per spike and capsule width, and also in number of spike per plant and average yield. Negative correlation is observed in between plant height and spike length, also in between number of spike per plant and spike length. So total 12 qualitative and 11 quantitative characters were computed and catalogued for morphological characterization. The natural cultivars exhibited substantial variability in morphological and agronomical characters except for 1 or 2 general traits. This is the dendrogram of large cardamom cultivars based on morphological data. This clearly indicates the Varlangi is clearly demarcated from other cultivars. And result of molecular characterization. The primary step in molecular characterization is the isolation of high quality DNA from different samples. So the DNA is isolated from the different cultivars. This is the electrophenogram view of genomic DNA isolated from large cardamom. The lane 1 to 10 represent the different large cardamom samples, and its quality is also checked using nanodrop spectrophotometer, and its purity is analyzed using A260/280 ratio and A260/230 ratio, and it is ranges from 1.5 to 2.6. And it's indicated that all the isolated DNA are in high quality, and total 20 ISSR and 10 SSR primers have been tested for molecular characterization analysis. The total number of amplified product was 238, and 145 bands. Out of this, 145 bands were polymorphic. That means 60.9% were polymorphic, and band size is ranges from 100 to 1700 base pair with an average 11.9 bands per marker. In total, 10 SSR markers are derived from transcriptome data and were screened and used to assess the genetic variation to discriminate cultivars of large cardamom. Out of the tested SSR, 6 produced amplification in all selected cardamom samples. However, the selected markers were more polymorphic for large cardamom. Is the amplification profile of ISSR and SSR marker, and figure 4 represents the amplification profile obtained on employing ISSR marker, different ISSR marker, and 5 is the SSR profiling of large cardamom. And this clearly indicate that ISSR markers are highly polymorphic for large cardamom, and SSR are always showing monomorphic banding pattern. The UPGMA-based cluster analysis correlation matrix clearly demarcated all the genotypes, and the genetic similarity matrix generated using ISSR data ranges from 0.45 to 0.88, and 0.88 is the highest and is observed in between Wolsey and Saramna. A similar result was also observed in PC analysis. It is clearly demarcated the outgroup varieties. So the present work is the first ever attempt made to evaluate and characterize the natural cultivars of large cardamom using different molecular markers. And the ISSR marker profiles different large cardamom accessions revealed a high degree of polymorphism between them. This indicated high levels of genetic diversity among the tested accession. Based on the morphological and molecular characterization data, the passport data sheets were prepared for all the genotypes. The first one is the passport data sheet for ICRS Sikkim 1, then ICRS Sikkim 2, Sami, Saramna, Wallangi, Wolsey, Ramsey, Ramla, Wild One, and ICRF 5. So the present study demonstrated the applicability of ISSR marker for analyzing genetic diversity studies in large cardamom. Extent variability was analyzed using ISSR molecular marker data, which enabled categorization of varieties at genome level, complementing conventional morphological data. And present study will be useful for defining descriptors for large cardamom. And the study gives ample scope to suggest that it will be useful for collection and conservation of germplasm, and also useful for breeding programs in large cardamom. These are some selected references. And I acknowledge DST, Department of Science and Technology, Government of India, and Spices Board for providing financial support and laboratory facility for this work. And I would like to thank Dr. Y.S. Rao for his guidance. And thank you all. I'm truly grateful for having the opportunity to participate in this event, and thanks to all organizers behind this fruitful event.
Our last speaker before the break is Anuradha Agrawal, who will give a talk on biotechnological applications for plant germplasm conservation at ICAR National Bureau of Plant Genetic Resources, India's recent achievements.
Day to participants in this session of Biotech 2021. It's my pleasure and privilege to give a presentation on some recent achievements related to biotechnological applications for plant germplasm conservation at the National Bureau of Plant Genetic Resources, India. The NBPGR is located in New Delhi and currently headed by its director, Dr. Kuldeep Singh. It has the vision to contribute towards sustainable development of agriculture by optimizing use of plant genetic resources, especially for adaptation to climate change, stresses, and changing anthropogenic needs. It houses the National Gene Bank, comprising seed gene bank, in vitro gene bank, and cryogene bank, which currently holds more than 0.46 million accessions belonging to 2,000 species of agricultural and horticultural crops. The Tissue Culture and Cryopreservation Unit at NBPGR houses the In Vitro Gene Bank and Cryogene Bank, where tissue culture and cryopreservation techniques are applied for the conservation of those species which are difficult to conserve in seed gene banks. The species prioritized for conservation in the in vitro and cryogene bank include vegetatively and clonally propagated crops, threatened and endangered plant species, crop wild relatives, non-orthodox seeded species, selected species of registered germplasm and release varieties, and voucher specimens of exotic germplasm. The major activities centered around the in vitro and cryogene bank include germplasm collecting and augmentation, health testing of the material collected or conserved, development of protocols for in vitro multiplication, development of protocols for in vitro cryopreservation, development of seed cryopreservation protocols including seed storage biology and basic studies, genetic stability assessment of conserved material, rejuvenation and retesting of conserved material, and evaluation and utilization of all conserved material. At the outset, protocols for in vitro multiplication and regeneration are developed for all clonally propagated and vegetatively propagated material, and most of protocols aim at no or less callus formation, that is, multiplication through direct regeneration, avoiding any hyperhydricity and somoclonal variation, and the protocols need to be applicable to a broad range of genotypes besides being cost effective. This slide shows a few examples of some of the recently developed protocols for in vitro multiplication in selected species. As an example, in vitro multiplication of a high medicinally important plant species, Hedychium, is shown in this slide. 17 different types of media were tested, and it was found that the response was genotype dependent. But subsequently, a single medium was devised consisting of MS salts with 4 mg BAP and 0.4 mg Na that was working across genotypes, though the response in terms of number of shoots varied. Micropropagation protocols are also sometimes refined with the objective of subsequent development of protocols for conservation, especially cryopreservation. And this particular slide shows the example of micropropagation protocol developed recently by our laboratory in the case of cardamom. Over the years, our laboratory has developed several in vitro slow growth strategies for conservation of germplasm. The more commonly used methods are those illustrated over here, and include minimal nutrient media as in the case of turmeric, use of mineral oil overlay as in the case of a medicinal plant Bacopa, low temperature storage in temperate species such as mentha, and use of osmotica in the medium as illustrated in the case of Campharia galanga, another medicinally important plants. These protocols have been published and have been widely used not only in our laboratory but other in vitro gene banks very effectively. Induction of storage organs in vitro is one of the most effective methods for in vitro conservation. And in our laboratory, we have successfully applied this in the case of ginger, taro, garlic, campharia. And once induced, these organs not only increase the shelf life of the cultures up to 3 years, but also can be used as a direct propagule for both in vitro multiplication as well as ex vitro multiplication. A combination of in vitro techniques is sometimes very effective method for in vitro conservation. This particular slide shows an example of in vitro conservation of Bacopa monnieri, a medicinally important species, where two slow growth strategies of reduced oxygen environment by use of mineral oil overlay and the synthetic seed technology was very effective for medium term conservation. And it was found that not only the techniques but also the explant can influence the storage of the cultures. And in case of Bacopa, nodal explants were found better than shoot tip explants for the medium term conservation of germplasm. This paper was also published very recently. While in vitro conservation strategies using slow growth offer medium term conservation, it is now important to develop long-term conservation protocols using cryopreservation. To begin with, I give the example of garlic. We have developed very robust and repeatable protocols for cryopreservation in garlic and other related species of allium as illustrated in the flowchart. And vitrification technique has been standardized using fresh cloves obtained from the field directly, in which buds have been isolated and subjected to vitrification solutions through a series of experiments determining the duration of PVS2, the duration of thawing and retrieval, and removal and regrowth media. To improve the recovery rates in cryopreservation of allium species, the droplet vitrification technique has also been successfully applied, which has higher rates of cooling and thawing. And using this technique, a large number of allium species, as indicated in the slide, including Allium albidum, chinense, hookerii, and several others, have been successfully cryopreserved with high recovery rates ranging from 20 to 80%. Cryopreserved shoot tips in most of these allium species have been not only checked for regrowth but entire plantlet recovery, and having checked the robustness of the techniques, most of the allium germplasm at NBPGR has now been placed in the cryobank. Genetic stability of the cryopreserved allium species has also been tested using morphological and molecular techniques, and the results show that the protocol developed at NBPGR has not caused any major genetic instability. Further validation of the allium protocol on cryopreservation has been tested by thawing 5-year-old cryoconserved material in selected accessions, and it was found that the data at the time of experiment and that after 5 years was almost equivalent in some of the cases, except few where it was either reduced or slightly increased. Droplet vitrification technique was compared with the vitrification technique for cryopreservation of shoot tips of Gentiana caro. It was found that the droplet vitrification technique could increase the recovery regrowth by almost a factor of 2 in case of this particular species, and therefore this has been used for cryobanking of this particular species. The in vitro cryopreservation protocol in Gentiana was also validated using molecular analysis by ISSR and EST markers, and it was found that no variation existed between in vitro cryopreserved plants and their respective controls. In our laboratory, we have been using droplet vitrification protocol for cryopreservation of Musa for a long period of time. But recently we carried out genetic stability analysis of cryopreserved material using in silico developed EST SSR markers. 40 EST SSR markers were developed and no significant difference among the in vitro cultured plants and the cryopreserved material was observed. We are also developing cryopreservation technique in Stevia rebaudiana using droplet vitrification, and so far about 60% recovery of whole plants has been obtained in this. Droplet vitrification technique has also been applied on Sisal microfilm with 35% regrowth, whereas vitrification protocol has been successfully applied for cryopreservation of Dioscorea rotundata. The encapsulation dehydration technique has been applied for cryopreserving hops, shoot tips, and so far only 40% post-thaw survival has been achieved and complete plants are yet to be recovered. Some species like Rauwolfia serpentina remain somewhat recalcitrant to cryopreservation protocols, and several attempts using both vitrification and encapsulation dehydration techniques have yielded only low recovery rate like 10%, and this needs further research. Cryopreservation using budwoods, especially in temperate species, has been a successful strategy, and it has been applied in the case of apple in our laboratory using two-step freezing with a good rate of recovery ranging from 11 to 40%. Dormant bud cryopreservation has also been successfully applied in the case of almond in our laboratory using two-step freezing. Protocol for seed and zygotic embryo cryopreservation of NSAID glaucum has also been standardized in our laboratory. We have demonstrated clearly that conserving either whole seed or excised embryos gives equally good post-thaw recovery. Similarly, ex situ conservation strategy for conservation of Musa bulbiciana seeds has been well demonstrated in our laboratory for conserving both in the seed gene bank as well as cryo gene bank. Our studies have clearly demonstrated the orthodox storage behavior of Musa bulbiciana, as they can be desiccated down to 5 to 10% moisture content. However, unlike other typical orthodox seeds, regeneration is only feasible through zygotic embryo culture under in vitro conditions. We have recently developed in vitro multiplication and cryopreservation in Garcinia indica, a underutilized fruit crop species with highly recalcitrant seeds, because seeds lose their viability below 30% moisture content. So in vitro derived shoot tips have been subjected to vitrification techniques, and almost 50% survival has been achieved after PVS2 treatment. It was also found that apical buds are better than axillary buds for survival after liquid nitrogen in case of Garcinia. In our lab, basic seed storage and conservation studies have been carried out on a large number of intermediate or recalcitrant seeded species as shown in the slide, and a substantial collection of these varied species are now conserved in our cryogene bank. Pollen cryopreservation to aid wide hybridization in different species has been undertaken, and this slide gives the example of Abel moschus species, where the pollen cryopreservation protocol has been standardized. Cryopreserved pollen has been tested for germination both in vitro and in vivo. And cryopreserved pollen of Moschatus subspecies Moschatus has been crossed with Abel moschus esculentus, and this wide hybridization has led to the successful fruit and seed set of hybrids. Over the years, more than 1,000 accessions have been monitored for retesting of cryobanked material, and viability has been retained in samples stored for 4 to 25 years. Rejuvenation and re-establishment of in vitro conserved material is also periodically carried out in our laboratory. And this example illustrates that in case of banana, where 24-year-old conserved material was brought in the field, it was flowered, and the material was established afresh in vitro. We have also established a very good protocol for rejuvenation of old cultures of taro, where endogenous bacteria is an inherent problem. This protocol, published recently, illustrates how fresh cultures need to be established in taro so that the latent endogenous bacteria problem is overcome. Periodically, we also undertake evaluation of in vitro conserved germplasm. This example is of the phytochemical diversity analysis of Lumbago zelanica, where one particular accession was found to be extremely useful in terms of its phytochemical content. Genetic diversity analysis of in vitro conserved curcuma germplasm using strap markers was also undertaken. And based on the dendrogram, two major clusters, the first of wild species and a few cultivars of curcuma longa, whereas the second cluster showed most of the curcuma longa germplasm was obtained. To conclude, the in vitro gene bank of NBPGR currently has 1,927 accessions belonging to 149 species of various horticultural crops, as shown in this table. And the cryogene bank at NBPGR has 11,936 accessions belonging to 825 species conserved in the form of seeds, embryos, embryonic axis, and pollen, as also dormant buds. Finally, I would like to bring home the point that our current research is focused on transferring germplasm from the in vitro conserved gene bank to the cryogene bank, and this table shows the success in the recent past where 291 accessions have been transferred successfully using various techniques. Thank you all for the patient hearing. I thank the organizers of this Biotech 2021 for the opportunity, especially Dr. Kanchit Tamasiri, and of course the director of ICR NBPGR, staff and students of TCCU who have contributed towards all the work that I have shown today. Thank you.
Our largest session, and we will have the Q&A joint session at the end of the of all 7 talks. But for now, we have a 1-hour break for lunch.
Just a quick reminder that you can visit the posters on the conference website, and we will have a slideshow of all the posters playing during the break.
After the break, Room 1 will continue with the plant diversity conservation, and Room 2 will finish up their session on physiology and production. So we will see you again at 1 p.m. Bangkok time.
Thank you.
Thank you.
Welcome back to room 1, where we will finish up with the last 3 talks on plant diversity conservation. Our next speaker is Miriam Kiadamashvili, who will give a presentation on the use of zygotic embryos for the efficient long-term conservation of a Georgian provenance of sweet chestnut in overview.
Hello, greetings to the conference hosts and participants. I'm Mariam Gaidamashvili, Associate Professor at Tbilisi State University, Georgia, and my today's speech is about zygotic embryos for the efficient long-term conservation of a Georgian provenance of sweet chestnut. Georgia is located in southwestern Asia between the Caspian and Black Seas. It borders Turkey and Armenia on the south, Azerbaijan on the southeast, and Russia on the north, with a total area of about 70,000 square kilometers. Also, it's a small country. Georgia features extremely diverse terrain, with both high mountainous ranges and fertile coastal lowlands. Most of the country is mountainous, with the Great Caucasus Mountains in the north and the Lesser Caucasus in the south. Georgia is one of the richest countries of moderate climate from the floristic point of view. Due to its uneven climate, altitude variation, and existence of ecological habitats. It is part of 36 biodiversity hotspots, the Caucasus and Irano-Anatolian hotspots identified by Conservation International as being simultaneously the richest and most threatened reservoirs of plant and animal life. Georgian flora counts more than 4,000 plant species. About 300 species of vascular plants are endemic to the country. 42 species of endemic flora are categorized as endangered due to disturbance to their habitats, excessive use, pathogens, and other pressures. Sweet chestnut, Castanea sativa, is a natural tree species in Georgia, covering about 4% of the country's forest territories, having high ecological, economic, and aesthetic values. Dendroflora of Caucasus lists 14 species within genus Castanea. However, only Castanea sativa occurs in Georgia. In addition to the common traits of biodiversity, such as overexploitation and climate change, chestnuts are endangered by a disease of high impact, as called edible chestnut blight, which is caused by the fungus Cryphonectria parasitica. This disease is listed worldwide among the most devastating plant epidemics. As a result, the large massifs of chestnut forest in Georgia are now on the verge of destruction. An official list of IUCN Castanea sativa has been assessed as a least concern. Also, it has been included in the red list of Georgia under state vulnerable. The low self-renewal of chestnut forests make an urgent need for the introduction of efficient contemporary conservation measures for both economic and wildlife protection commitment. The establishment of the chestnut plantations using conventional techniques is problematic because chestnuts are very difficult to propagate by vegetative reproduction. As to ex situ conservation in seed banks, the seeds of Castanea sativa are classified as recalcitrant, in other words, non-orthodox seeds, and not suitable neither for planting nor for seed banking. Cryopreservation as the safe and effective method for long-term conservation of different biological materials proved to be an exceptional choice for the preservation of threatened species. Several recent works have contributed to the maintenance of Castanea species and hybrid clones at ultra-low temperatures for an unlimited time using different explants such as shoot tips, zygotic embryonic axis, pollen, and embryogenic cultures. The success in conservation strategies applied to the threatened species largely depends on elaboration of efficient in vitro propagation methods of particular species. This is because in vitro methods are essential for the successful recovery of explants after cryopreservation. As a general rule, younger tissues like zygotic embryos have better potential and competence to produce organs compared to more differentiated and mature tissues. Other benefits of zygotic embryos are attributed with metabolite profile and differences found between zygotic and somatic embryos, where 40 times higher proline content was detected in zygotic embryos compared to somatic ones. Besides, zygotic embryos contain significantly higher contents of sucrose, gamma-aminobutyric acid, and alanine, which play an important role in acquisition of desiccation tolerance. Seed morphology of hardwood species is often an additional reason for the preferred choice of excised embryonic axis for long-term conservation, because various tree species such as chestnut, walnut, oak, and several tropical species have large cotyledons that make the seed size not suitable for using cryopreservation. In this case, there is a possibility to store excised embryonic axis in liquid nitrogen after the application of appropriate dehydration protocol. From the conservation point of view, axis and cotyledons of recalcitrant seeds of Castanea sativa exhibit contrasting responses of respiration and sensitivity with desiccation, and their conservation at low above freezing temperature allows the maintenance of seed viability and germinability for very limited time. As to cryopreservation, the desiccation method has been broadly used for the preservation of zygotic embryonic axis of chestnut species and hybrid clones. Cryopreservation protocols elaborated for the Georgian provenance of Castanea sativa in the tissue culture laboratory at Tbilisi State University in collaboration with the Institute of Bioeconomy in Florence have been useful in achieving a successful conservation program for this species. Experiment on one-step freezing cryopreservation of the embryonic axis from mature seeds using different techniques resulted in inducing specimen tolerance to ultra-rapid freezing. For cryopreservation experiments, the fruits were washed, then the seed coat and lateral part of endosperm were removed. The remaining were self-sterilized and excised embryonic axis were desiccated in open petri dishes under the airflows of a laminar flow hood for different desiccation times from 0 to 5 hours. Embryonic eggs were placed in 2 milliliter cryo vials and immersed in liquid nitrogen for 24 hours. For thawing, the cryo vials were rapidly immersed in water baths at 40 degrees. After thawing, samples were cultured in VPM medium with 0.4 benzenamylopurine and 0.3% activated charcoal in culture tubes and were maintained in growth chamber. After 5 hours of dehydration, maximum survival in post-cryopreservation achieved by embryonic access was 80%, followed by plantlet regrowth at 66.7%. Besides the high germinability obtained with the dehydration cryopreservation procedure, the germinated embryonic axis could be easily acclimatized and produced healthy seedlings without any signs of morphological alteration or callus proliferation. Also, the PVS2 vitrification protocol developed in the present study showed a good applicability to the Georgian provenance of chestnut, although to a lesser extent than the dehydration one-step freezing technique. Chestnut embryonic axis tolerated well the PVS2 treatment in terms of early germinability, as called survival, but less in terms of further germination, as called plantlet regrowth. As a consequence, PVS2 vitrification solution for 30 minutes produced 55.6% of fully developed plantlets in post-cryopreservation, which was the best result for this technique. Encapsulation vitrification technique as an alternative to desiccation and PVS2 vitrification one-step freezing has also been tested on Castanea embryonic axis. This method combines the advantages of vitrification and encapsulation of explants, greatly reducing the treatment time in comparison to encapsulation dehydration method. An improved cryopreservation protocol has been developed for the embryonic axis, including 0.3% of activated charcoal in alginate beads. Cryopreserved embryonic axis with activated charcoal showed 70% embryo survival and 64% recovery into the whole plants, which was sufficiently higher in compared to those of without activated charcoal. Apparently, the inclusion of activated charcoal in alginate beads has eliminated the toxic effects of phenolic exudation, which is an important issue in chestnut micropropagation. Also, Castanea sativa is the most studied chestnut taxon for micropropagation and conservation worldwide. The Georgian provenance of Castanea sativa was originally studied for conservation by cryopreservation. Our researchers demonstrated the feasibility of long-term preservation of embryonic axis using different techniques such as dehydration, PVS2 vitrification, and encapsulation vitrification, followed by one-step freezing in liquid nitrogen. The practice can be generally implemented to a wide range of chestnut cultivars and hybrid clones to achieve the practical long-term cryopreservation of Castanea genus germplasm. At the same time, current work offers a useful platform for further work towards the improvement of the conservation strategies applied to the threatened hardwood species that cannot be preserved conventionally, thus fulfill the phytogenic pool of the country. Thank you very much for your attention, and I hope I will be able to answer your questions. Thank you very much again.
Our next speaker is Kanyaporn Pipit Sangchan, who will give a talk on safeguarding and use
Hello,
of plant genetic resources in DOA Gene Bank.
hello everyone. Today I'm Kanyaporn Pipisanchan. I'm going to talk in the topic of safeguarding and use of plant genetic resources in the Gene Bank in case of Atitayathon Agricultural Project in contemplation of Her Royal Highness Princess Atitayathon Kittikul Atsurin has collaborated with DOA Gene Bank execute conservation of some field crops and horticultural crops in the in Bank, Thailand, 2019 up to 2020. This is the Kingdom of Thailand. We are in Southeast Asia, and Bangkok is our capital of Thailand. And this is the area of our research. Atitayathon Agricultural Project in contemplation of Her Royal Highness Princess Atitayathon Kittikul at Surin Province in lower northeastern part of Thailand. And this is DOA Yin Bank at Pathum Thani Province in the central of Thailand. And this is Her Royal Highness Princess Atitayathon Kittikul. She is the one who grant the name of this project. And she is also the second daughter of Princess Juraporn Varairak. And this is the pictures of Gene Bank, the building of Gene Bank Research and Development Group, Biotechnology Research and Development Office, or we called DOA Gene Bank. And this project, we also collaborated with Surin Agricultural Research and Development Center in Surin Province as well. This is the capacity of DOA Gene Bank. We have 32,977 accessions and 184 species. This is the medium-term storage room inside the Gene Bank. We have 5 degrees Celsius and 60% relative humidity, which can conserve the seeds up to 20 years. And this room is about 86 square meters with 24-meter height. Its capacity is about 150,000 successions. An automatic bullet crane system is used. PET plastic bottles is used. And this is long-term storage, minus 10 degrees Celsius. It can preserve the seeds up to 50 years and up. And this is room. This room is about 76 square meters. Its capacity is about 40,000 accessions, vacuum aluminum foil package. These pictures showing Celer Atitayathon Agricultural Project. Her Royal Highness Princess Atithyathorn Kittikul granted the name of this project. The internal area of the project is zoning for the activity for promoting agricultural learning center where the farmers of the community can take advantage for sustainable agriculture, including education, knowledge, and technology as well. Normally, in the project area also have preserved the seeds for the farmers and distribute seeds to the farmers in the next season for planting. Department of Agriculture, by the way, Yin Bang and the Sla Atithyathorn Agricultural Project concerned about the significance of sustainable use of the preserved seeds. Therefore, the collaboration between the Yin Bang in the process of the PGR conservation and Yin Bang management and seed deposit in the Yin Bank in long-term storage had been conducted. The objective is the first, duplication and back up the seeds. The second, increasing PGR diversity. The third, assurance for the food security, including conservation and sustainable use for plant genetic resources. The fourth, promotion of the agricultural research and bring to utility from the plant germplasm for the farmers. For maturing methods, collection and plant germplasm, seed laboratory and seed storage, seed characterization, and evaluation. Collection the plant germplasm. 12 plants such as holly basil, chili, sweet basil, red chili, turkey berry, eggplant, lollipop, fiber, orchiga, rice, green oak lettuce, red oak lettuce, yellow velvet leaf or pak pie, and dill. For holly basil ossimum tenuiflorum, we have red and green. For chili, capsicum annuum, sweet basil, we have ossimum basilicum. Bird chili is our capsicum frutescens. Turkey berry or มะเขือพวง Solanum torvum, and eggplant or มะเขือเปราะ Solanum pilongina, and also lollipop, fiber or Kika, Bryonia lacinosa, and rice, Oryza sativa, green oak lettuce, Lactuca sativa, and red oak lettuce, Lactuca sativa as well, and yellow velvet leaf or pak pie is Limnocalis flava and Anitum glaviolens. This is the seed laboratory and seed storage process inside the Yin Bank. We gather the seeds from the area and then bring back to the laboratory. And then we clean the seeds for the seed characterization. For chili, capsicum annuum, and also bird chili, capsicum frutescens, we do in-house method description for capsicum. We use IPRI 1995, and also the details of inspector for the new plant variety as Plant Protection Act 2542 BE, and it's belonging to Department of Agriculture. And also Ceylon spinach or Bacilla agar, tomato, Lycopodium esculentum, and also Thai basil and holly basil. And this is the features of the products inside the area. for chili, we have 4 locations and 12 rows per variety and 4 locations. Plant density about 50 times 100 centimeters. The morphological characteristic data were collected. 67 parameters. The seedling development stage is about 4 parameters such as hypocotyl color, hypocotyl pubescence, color of cotyledon, shape of cotyledon. The vegetable growth stage, we have 13 parameters: stem color, stem shape, stem pubescence, plant height, plant canopy wise, and plant growth habit, leaf density, leaf color and leaf shape, laminar margin, leaf pubescence, length of mature leaf. And for eggplant, we also conduct for the morphological characteristic data were collected about 50 parameters. This is the morphological characterization of eggplant in vegetative growth stage. And this is in flowering stage. We have day to flowering, number of flowers per inflorescence, number of hermaphrodite flowers per xylem, and pollen production, relative style length, pedicel thickness, fruit pedicel prickles, corolla color. And we also have morphological characterization of eggplant in fruiting stage and also in seed stage. We have the pictures of leaf form, leaf hair, and leaf hair in dorsal surface, flowers, flower color, immature fruit, mature fruit, and mature fruit cross-section. For Ceylon spinach or Bacilla alba, we also have tomato, Lycopersicum esculentum. These pictures of tomato, and we characterize Holybaceus, which green and also red one. Morphological of Holybaceus in vegetative growth stage. The days to germinate is 7, and hypocotyl color, intensity of hypocotyl color, stem color, plant growth habit, plant height, growth rate, leaf shape, leaf color, thickness of mesophyll. And this is, for example, of Holybaceus in the productive growth stage. And this is Thai basil or Horapa oxymum basilicum. And rice also do. We characterize rice in the Oyin Bank, Pathum Thani province. And this is a picture of seed bank of solar agricultural project at Surin province. For the rice seed, we conduct the research in the DOE in Bank in Pathum Thani province. In long-term storage room, we collect the seeds of this project in this position of this room in our gene bank. And this is the use of plant germplasm of the project. We have eggplant, yellow velvet leaf, or pak pie, Ceylon spinach, bird chili, turkey berry with chili paste. And we also have stir-fried pork with Holybaceus and red and green one with chili. It's delicious one too. And we have green oak lettuce and red oak lettuce and tomato. We also have some chicken curry with sweet basil, eggplant, chili, dill. And this is our seam rights. At last, no poverty and zero hunger will occur around this area in the near future. Thank you for your attention.
Thank you. Our last presenter of the session is Tripachara Patiratana, who will give a talk on comparative assessments of alkaloids and phenolic compounds in a Thai medicinal plant, Ericibe elliptilimba, and other species in the genus.
Hello everyone, I'm Tripachara Patiratana. Today I would like to present my research article on the topic of comparative assessment of alkaloid and phenolic compounds in a Thai medicinal plant, Ericibe elliptilimba, and other species in the genus. In this presentation, I will start with background and objectives of my research. And the next, I will talking about material and methodology, resource and the discussion. And finally, I will conclude and summarize my research. First, I will talk about my research background. Ericibe elliptilimba, or Thai medicinal plant named Pakhanchaisi, is a plant in the morning glory family or the Convolvulaceae. The stems of this species are used on the Thai traditional medicine for treating unknown fevers such as malignancy. In addition, in vitro study, the stem of Ericibe elliptilimba have the potential of the anti-proliferative ability. For the phytochemical study, these species are found two phytochemical groups, including one coumarin that have the structure one benzene link and the one heterocyclic link. One compound in coumarin is the scopolectin, and this compound has the many potential in the traditional using, for example, anti-inflammation, treating convulsion, and treating rheumatic pain. And another phytochemical group is the topane alkaloid. They have the hydrocarbon containing the amine group. They commonly found in the Solanaceae and Convolvulaceae. And interestingly, callistogen is the one of the topane alkaloid and only found in the Convolvulaceae. Moreover, in the conservational study, this species were recorded as the least concern in conservation status, but the frequent collection of this species stem from medicinal using can affect or lead to the risk of becoming a threatened plant species. So, to decrease the risk of the threatened species, Cunningham suggests the two strategies for the sustainable use of the medicinal plant. For the first strategy is discovering effective alternative species. For the example, they want to find the alternative Ericibe smithii by testing the pharmacological activity, and they found that Polana sinases have the same pharmacological activity comparing with the Ericibe smithii. So the Polana sinases could be as the candidate for the alternative Ericibe smithii. And the second strategy is the finding alternative plant path, because they want to decrease use of the ginseng root. So they compare the ginseng root with the another organ, including leaf and stem. And the results show that all of organs that they test show the similar activity as the root. So leaf and stem of the ginseng can be the alternative plant path for the ginseng root. And all of these can lead to my research. Elizabeth Limba will be find alternative by considering the Cunningham strategy for the first discovering effective alternative species and the stem of the T species including Elizabeth albida, Elizabeth cytinifera, and Elizabeth staphyena will be compared with the stem of the Elizabeth limba. And the second strategy is the finding alternative plant. The leaf of the four Elizabeth will be comparing with the stem of the Elizabeth limba for the finding alternative. But how we know that they have the potential for the alternative? The one of method they were used for the screening alternative is the histochemistry. The histochemistry is the knowledge that integrates biochemistry, anatomy, and the physiology. And the result they give the qualitative result and they have the benefit which is the localization, simple and economical. So that's all of this that lead to my objective. For the first, to use histochemistry to screen for phenolic compound and alkaloid in the stem and leaf of 4 species in the genus Elizabeth. And the second, to find other candidate species as well as alternative plant organs that have the similar chemical compound as the stem of Elizabeth limba. And this is my overview of my research. First, the Elizabeth were collect and identify, and then histochemical technique will be used for the finding alternative. Elizabeth was surveyed and collected, and after that, whiter specimens were prepared by Bryson and Foreman method. After that, whiter specimens were identified by the flora of Thailand, China, and neighboring countries with the considering by experts Mr. Pongsakorn Kongchaiwas, and after that, the whiter specimen deposited in the BKF. And this is the species that I concern in my research. The fourth species, which is the Elizabeth limba as the medicinal plant, and Elizabeth albida, Elizabeth cytinifera, and Elizabeth staphyena for the common species. And the next method is the histochemistry. The first stem and first leaf will be sectioned by sliding microtome or the handing section, and then they stand in the two chemical staining reagent, including first natal stop for phenolic compound, and second is the Dargendorf reagent for the alkaloid and topane alkaloid. And after that, the results were observed and photographed by the light and fluorescence microscope. And this is the result from the table. First column is the species, and in each species have the alphabet referred to the picture from the right hand. And the next column is the staining reagent from the natal stop for the phenolic compound, and Dargendorf reagent lying in the sodium nitrate for the alkaloid and the topane alkaloid. And the result found that in the bark, Elizabeth albida and Elizabeth cytinifera have the same positive reaction comparing with the Elizabeth limba. And the next, Elizabeth albida and Elizabeth cytinifera also have the positive reaction comparing with Elizabeth limba when standing in the root, and this result can support in the first strategy of the Cunningham that discovering effective alternative in order to reduce destructive harvesting. And Next is the histochemistry of the leaf of Elizabeth, and the result found that Elizabeth limba leaf have the positive staining reagent similar to the Elizabeth limba stem. And these results are necessary to the Cunningham strategy for finding alternative plant paths. From the result, which had the different staining reagent of the Elizabeth, they can explain by many research. For example, Earthman explained this event by the difference may be due to small changes caused by mutations affecting the production of secondary metabolites. And Earthman and World Health Organization also says about the environmental factors such as soil condition, season, and climatic factors can also cause variation in the secondary metabolite product. And in addition about the result, World Health Organization said that the preliminary results should be support the more sensitive instrument to develop chemical fingerprint and isolate tests to discover active ingredient or the contribute the active marker. And the next author they says about the phytochemical compound do not necessarily to equally to the pharmacological activity. So if you want to the strong information, this species should be tested for the pharmacological activity related to the traditional uses. And there's some suggestion that is that collect plant identifications such as micromorphological character, macromorphological character. Also, the molecular approach is the key to controlling and supporting the quality of medicinal plants. So that all of this can conclude that the stem of Elizabeth elliptilimba have similar phytochemical compounds as the stem of Elizabeth cytinifera and Elizabeth albida. And the second is the list of all these alternative species have the same phenolic compounds as Elizabeth elliptilimba, but the alkaloid results reveal some difference from the four species. Finally, I would like to acknowledge Science Achievement Scholarship of Thailand for funding my research and co-author Dr. Pavina Taiperm, Dr. Thanika, and Dr. Ashila, also Mr. Pongsakorn Kongchaiwan for support this research, and also Ms. Kanchana Pamali for supporting and suggestion, and also Pan Taxonomy and Phytochemistry member. Thank you for your kind listening.
Okay, this concludes all of the talks for plant diversity conservation. And we will now have a joint Q&A session for all 7 speakers in this section. In this session.
To start off the Q&A session, I have a question for Sandhya Gupta. If you are here, can you please unmute your microphone? My question for you is, based on your experience and studies of similar species, how long do you expect cultures of Artocarpus lakochek can be maintained?
Those cultures which has been conserved using different strategies, I found normal growth strategy is the best one, and cultures could go until for a year. So far, the experiments are going on, and for 1 year now, the cultures are all fine. But many top cultures can go a little longer, but we have to see there is any abnormality present or the cultures are all normal as a mother culture. So yeah, the experiments are going on. Yes.
Okay, we look forward to hearing an update perhaps at next year's conference.
Yes, of course.
Thank you, Sandhya. I also have a question for Wichai Buri Banya Wanit. If you are here, could you please unmute your microphone? My question for you is, what is the distribution of Nymphaea siamensis? Is it found throughout all regions of Thailand, and is it found in any neighboring countries?
Are you with us here, Vichai?
Okay. Do you listen me now?
Yes, we
Yeah,
can
we
hear
hear.
you.
Yes. From the history of Thailand, we have the Nymphaea siamensis or Jongkoni water lily. In addition to my explorer in Thailand, but this is secret. I also cross the border to Laos or to Myanmar or some other neighboring country. We can say that Nymphaea siamensis is the unique of Thailand. Okay.
And
And
a
for
question from me. Do you think Nymphaea siamensis might be derived from other species, maybe through polyhybridization or hybridization or other event?
Of course, but we should say that it's not derived from the other species. We should say that it's the one of the evolution or mutation from mutant from the other species, right?
We also have
So,
a related question from Anuradha Agrawal, who asks, what could be the possible causes of absence of floral organs and its evolutionary significance? And is it also known in other species within the genus?
Yes, for my presentation, you should see that please see the uh, the principle of the taxonomy is in this genus into two group of six subgenus. One is uh, Apocarpi and the other one is Syncarpi. But Jongoni water lily or Nymphaea siamensis have no any locule how how to provide it in any group, right? Okay, do you listen to me?
okay,
Yes,
thank you very
thank
much.
you for your answers.
Yeah. And we have a question. We have questions for Anuradha Agrawal. Are you here with us? Can you please unmute your microphone? The first question from Dr. Thamasiri. He would like to know, did you try using V-cryoplate and D-cryoplate for cryopreservation of some crops? And do you cryopreserve jackfruit and how? Sorry, we cannot hear you at the moment. Maybe we have some technical difficulty here. Yeah, we still cannot hear you. Actually, I think you can just type in the chat box to answer the question. I will have my staff to provide the question in the chat box, and you can answer there.